In this lesson, we are going to learn about an important component of bacterial cell structure called cell envelope. As you can understand, this is the outermost protective covering that serves to protect the cell from its outer environment. Bacterial cells are surrounded by this cell envelope, which is typically made up of 2 to 3 layers depending on the bacteria. One of these layers is called cell wall, which is composed of a substance called peptidoglycan. As the name indicates, peptide refers to some protein structure, glycan refers to some sort of carbohydrate. So peptidoglycan is made up of protein and carbohydrates linked together. There are 3 types of bacteria based on their cell envelope, namely gram-positive, gram-negative, and atypical cell wall bacteria. This information is used in medical science to determine the course of treatment when we know what type of bacterial infection a patient has. Is it gram-positive, is it gram-negative, or it's the third one? What is the basis of this difference in cell wall structure? Well, in gram-positive bacteria, which are represented by blue color in this image, you could see that the cell envelope is composed of cell wall, again made up of peptidoglycan, and plasma membrane. In this image, you are shown the detailed structure of peptidoglycan composed of these thick blue cylinders representing the glycan sheets, which are linked together by these thin blue fibers representing the peptide, the polypeptide chains that link the glycan sheets with each other. If you notice, there are several layers of peptidoglycan in the gram-positive bacterial cell wall. Cell wall is followed internally by plasma membrane, which is a phospholipid bilayer integrated by various proteins that aid in transportation of material across the plasma membrane into and out of the cell. This other type of cell here, which is represented by red color, is gram-negative bacteria. So if you look at the gram-negative bacterial envelope, it's composed of 3 parts: the outer membrane, a peptidoglycan layer, and plasma membrane. So if you compare the 2 cells, you see right off the bat that the outer membrane is missing from gram-positive bacterial cell wall. That's number 1. Number 2, you notice the peptidoglycan in gram-negative is very thin. It's extremely thin, just 1 layer of peptidoglycan. In gram-positive, there are several layers of peptidoglycan. So what are the 2 main differences to remember? Gram-positive, no outer membrane, thick peptidoglycan. Gram-negative, outer membrane, thin layer of peptidoglycan. Now, I want to talk a little bit more about this outer membrane, which is made of this substance called a lipopolysaccharide. And if you break down the term, you will understand that it's composed of this complex molecule is composed of lipids and complex sugars, polysaccharides. You should also notice before we move on to the next slide that plasma membrane is the same in both types of these bacteria. So the main difference comes down to the presence or absence of outer membrane and the thickness of peptidoglycan. But I would also like to note before I move on to the next slide is that these 2 colors are basically symbolic because they represent the colors of bacterial cells at the end of a special staining procedure called gram staining, which is used widely to distinguish bacteria into these 2 groups. So let's quickly review. Gram-positive cell envelope is composed of thick cell wall of peptidoglycan, inner cytoplasmic membrane. Gram-negative cell envelope is composed of outer membrane, thin cell wall, only one layer of peptidoglycan, and an inner cytoplasmic membrane. The third type of bacteria we mentioned earlier, the atypical bacteria, they lack a cell wall, a peptidoglycan cell wall altogether. These cells are only surrounded by a plasma membrane, which is further strengthened by the presence of these organic substances called sterols, which give some additional protection to the cell's environment. As I mentioned, I would like to talk a little bit more about this lipopolysaccharide, because that is the stuff that acts as endotoxin responsible for one of the major illness that is caused by gram-negative bacterial infection. So lipopolysaccharide is a complex organic molecule made up of lipids and sugars. Lipopolysaccharide make what is referred to as the outer membrane in gram-negative bacterial cell envelope. This substance acts as endotoxin in case of a gram-negative bacterial infection and can lead to a life-threatening condition referred to as endotoxic shock. Lipopolysaccharide is also recognized by our immune system, the system that helps us fight infections, as a marker for the detection of bacterial pathogen invasion of our body, responsible for the development of inflammatory response, and in case of a severe infection, when there is an extremely high amount of endotoxin present in our system, it results in endotoxic shock. Now, this difference in bacterial cell wall that we talked about in terms of the amount of peptidoglycan, the presence or absence of outer membrane, it resulted in development of a special staining technique that is called Gram's staining, developed by this scientist by the name of Christian Gram, used for microscopic study of bacteria, which delineates them into two major groups. Gram-positive bacteria, the one with thick cell wall, remember? And gram-negative bacteria, the one with only one layer of peptidoglycan. Please follow the link below to watch animation on the Gram staining procedure. You could type this in your browser. This is a technique that we will also be practicing a lot in our lab.